Hello everybody, good morning. Welcome to the Metabolomics Summer Workshop 2015. My name is Mathew. I'm a clinical instructor in the Division of Nephrology. You probably had looked at study design, learned the nuances of it, but I'm hoping to convince you to look at it in a metabolomic standpoint, and so that you can fine-tune your study design methodology to get the results you are hoping to get out of this. So you have an idea, and you're thinking about using metabolomics to answer your question. I highly recommend this aspect that Chuck mentioned earlier, that is consultation with the core. Because there are a lot of things that go unnoticed in study design preparation. Sample reaches the core, the answers are given back to you, and you look back and think, oh, I should have thought of that. So to avoid those kinds of faux pas, I think it's a good idea to have consultation with the core. In this talk, I hope to convince you how to answer your biological questions with metabolomics, and what are the factors to consider when you design a metabolomic study. So initially, we'll talk about the different model systems that you could use, biological materials that we can work with. We'll talk about different study designs and the power needed to answer your questions. We'll talk about this study design factors that alter the metabolome that might be really important to your study design, and strategies to prevent these variabilities. So you have a question that needs to be answered, and this question can be hypothesis driven. You have a hypothesis, you're interested in certain parts of the metabolome, be it lipid, carbohydrate, amino acid. You have a panel of metabolites you are interested in, there's a pathway you believe is altered. The best way to answer that is through a hypothesis driven targeted approach. The other approach is you have a well-defined cohort, you have a well-defined difference that you want to figure out. A hypothesis generating approach called untargeted metabolomics is the way to look at it. The way I think about it is targeted approach is really looking closely at things. You know what you're looking for, you get a quantified answer. For the untargeted approach, it's a bird's eye view of things. You measure a lot of variables, and differences that are huge are readily apparent. And sometimes you can lose minute details and minute changes that you're really not looking closely at. Therefore, you need a combination of both, or if you have a real good hypothesis, a good targeted approach would answer your questions. Now we'll talk about the different model systems. The simplest and easiest to do probably is a cell study, where you can decide what endometabolome, what intracellular changes are happening, causing metabolomic perturbations. We can also look at the exometabolome, where you can look at the extracellular milieu, in this case the cell culture media, to see what's going on that's changing between your experimental conditions. This is very reproducible. We can use small numbers to get the condition. All the environmental conditions are in your control, which could be a good thing or a bad thing, because small changes that you make in your study design would make huge changes in your answers. This is also a great platform to start studying flux of metabolites. You can introduce isotope labeled flux. These are metabolites with small changes in the amount of mass changes that we can detect through mass spectrometry. This can be tracked to different pathways and gives us tremendous information about the metabolome that's happening in the cell. Second model system is the animal studies. These are another great way to look at things. A lot of interventions can happen in the system. A lot of inter-individual variations are minimized because these are all usually from one single genetic background, and you are controlling the diet and the environment. Therefore, you have well-defined differences between the cohorts. The other thing that this allows us to do is to do simultaneous harvesting of plasma, urine, tissue. Therefore, it gives you an entire picture of the model system. Human studies, of course, the genetic background and the environmental background is extremely diverse. This accounts to many of the changes. The biological variability in individual samples are going to be high, but adequately powered studies will give you great answers. Also, the good thing about this is nearly 4,000 metabolites have been quantified and published in human plasma and human urine that are quantified ranges available. Therefore, this is highly studied, and therefore things can be referenced back to normal values. So there are varied biological materials that we can work with. With the cell system, we typically require 1 million or 10 million cells per plate to get you a good answer. We can also assess the culture media. For animal and human systems, we can assay the plasma, serum, urine, saliva, CSF pleural fluid, ascitic fluid, tissues like skin, nerve, and various organs. And now that we are looking at the metabolome as a composite of microbial metabolome as well, fecal material, which contributes a lot to the human metabolome, can also be assessed. So what study type is good for metabolomics experiments? So case control studies, these are usually drawn from the population, well-defined cohorts with a well-matched control is ideal for studying metabolomics. They usually have excellent correlation to the disease studied. They reveal altered pathways. They have a biomarker potential if they are adequately powered to detect differences. But the cons are these are prone to selection biases from population. Also, the issue of reverse causality happens. You saw a metabolome change with an intervention. Is this the cause of the intervention, a result of your phenotype? That's usually hard to tease out, but we can design a good study that can kind of tease out these things for you. There are also study design artifacts. For example, if you're studying patients with heart disease and patients with heart disease and without heart disease, and you see drug metabolites of statin in one group, that is a study design artifact because obviously the people with heart disease are on statins, therefore you see their metabolites in the serum. Therefore, there are some study design artifacts that crop up with these kinds of studies. The second kind of study is an intervention study where you can have time series data between intervention before and after. A time series data gives you information on the stability of the metabolites. Therefore, you can solidify the presence of these metabolites as a biomarker for future prospective things. Intervention could be either from change in phenotype or change with the intervention itself, and that needs to be really defined. Crossover studies are best strategies because you eliminate the problem of inter-individual variations between the subjects, because these subjects go through both placebo arm and active treatment arm. Therefore, the same controls can be controls for the treatment arm as well. Therefore, you are eliminating the inter-individual variations. So the question we always end up with, what is the sample size that's required to get these answers? What will achieve the right power? You need this to put it in grants. This is a very wild question. You can spend a lot of money and not get the answer you need if you don't think about this. So compared to normal experiments that we use in science, metabolomics is slightly different because we have the capability to measure thousands and thousands of variables. And in target approach, we have a limited panel still. We have a lot of variables that we measure at the single time. What does this do to us? So this interferes with the statistical inference of things. Multiple variable measurements increases the false discovery rate. The amount of false positive increases the ability to, this causes an increase in the type 1 error of a study. Therefore, we have to use some statistical method to control for this. Therefore, we come up with a stringent cutoff to limit the false discovery rate. A stringent cutoff has two things to do. It means it needs more sample size to get the same power that you want. And it also means that because you're measuring multiple things at a time, valuable data can be suppressed in the stringent cutoff. One way to get through this issue is to do a discovery study or a pilot study that we talked about. Use a small cohort of matched controls and case studies that we find what the effect size is. Based on the effect size that you see, you design a larger, well-powered cohort that is adequately powered to answer your question. Biological variability, especially in human studies, is an issue. Sometimes heterogeneity between the samples cause larger variation. This can be short-term variations, as in you sample the samples at different time points, therefore you have different answers. Sometimes you sample things across long-term time. For example, vitamin D levels in the blood changes over summer and winter. Therefore, you need to account for all these things. Again, this needs higher sample volumes. Before planning a good metabolomic study, it is necessary to plan for data normalization. So you're going to measure all these things. Are you measuring equal number of things? One way to get around things is to count cell number in cell studies. As I told you, 1 million to 10 million cells. If you are consistent throughout the entire experiment, you get the answers you need. Sometimes you can decide it is a ratio of the metabolite that you equally distribute across the experiment. Many biological fluids you can decide to use equal volume. For example, plasma, serum, these are at good homeostasis in the body. You can use a certain volume of the sample. Certain biofluids like urine and plasma, if you're measuring things that change on, quite a bit during the day, can use key metabolites like glucose to normalize for the data. In urine, a lot of concentration changes happen, and we nephrologists typically use creatinine in the urine because it's a steady measure every day to account for the day-to-day changes in the concentrations of urine. You can also use urine osmolality as another measure. Tissue samples can be normalized using protein concentration at the outset, which means before you give the sample to extract to us, you decide to do a protein concentration measure, because sometimes after the fact doesn't work. So these needs to be considered when you do a study design. Sometimes dry weight of the tissue is more ideal, especially things like fecal material. You might need to do the dry weight of the fecal sample to see that you have the same amount of sample across the board. Sometimes you have not thought of this before. You can try to use the total ion chromatogram the machine generates as a way to standardize for your answers. So what are the factors that affect the metabolome? At this point, I want to pause and make you realize that we are capturing a snapshot of the metabolome at most times, and we are not capturing a movie. So you're capturing still pictures at different points, and we need to make sure you're capturing at the right moment. So timing is paramount. So for example, in the cell metabolome, when you are doing the experiments, you decide how much seeding you do, the densities matter. At what time are you going to capture the metabolome? Is it at the proliferation phase, at the growth arrest phase? How many lifespans have all your experimental cells gone through? Are they equal across? How many passage numbers have they gone through? When do you decide to harvest these cells? For example, in the graph here, these are myeloma cells that we have measured glucose and lactate. They're two different media, they're for the different colors. As you see, the glucose level drops over time because they're getting used up by the myeloma cells. While that happens, the lactate levels go up in the cell culture media. So where you decide to interrogate the metabolome changes the picture totally, as you can see. So timing is important where you decide to sample a cell metabolome. Also things like temperature, ambient atmosphere make huge changes to the proliferation capacity of cell cultures. Therefore, you need to be consistent across the board. Culture media composition cannot change across experimental conditions because nutrients like glucose, glutamine determine the proliferation aspect. And the only thing that can change across the experiment is the intervention that you're trying to measure as. Also things like additives that may sometimes suppress a chromatogram needs to be considered. This is where consultation is a good idea before you plan these experiments so that you're not doing back thinking after you do it. This is one more example of timing. In this case, this is a human experiment of 10 healthy volunteers who are made to stay awake for 40 hours, and we are measuring the steroid precursors, the long chain fatty acids in this case. As you can see, as time goes on, these steroid precursors show a very definite pattern. And at 24 hours, if you draw a line, you can see that the pattern seems to be repeating itself. The same pattern repeats again. This is because of the circadian rhythm our body has. So again, timing in this situation is very, very important. Where you do decide to draw the blood. Do you draw the blood first thing in the morning? And this is the picture you'll get. Later on the day, you'll get this picture. So you have to decide what is valuable for your experiment, where do you want to draw the blood, and what kind of answers you're trying to answer. So timing of collection needs to be standardized and cannot be changed throughout the study. It has to be fixed at one point and stuck to along. So what are the other things that change the metabolome? Other than the body's circadian rhythm itself, fasting states, feeding states, activity, stress, all have changes to the metabolome. Here I'm showing an example of a study with healthy volunteers where we are measuring free carnitine and acyl carnitine. The red line is the free carnitine and the green line is acyl carnitine. The first few hours, first 24 hours, they are fasting. Even in the fasting state, you can see how the metabolome changes over time. So even if you decide to draw your sample in the fasting state, you have to fix the time point of fasting. Either it's 8 hours or 12 hours and stick to it throughout the study. Here you can see the differences glucose tolerance test does to the metabolome, specifically carnitine in this point. This is a lipid challenge, as you can see. Here, the patients were undergoing an hour of treadmill exercise, and you can see how that changes the free to acyl carnitine levels. Also, you can see here, they made the subjects immerse their hand in cold water for 3 minutes. That's all they did. That's a measure of cold stress, and you can see how the metabolome changes in response to 3 minutes of cold stress. So your study design needs to take into account what you're trying to achieve and keep it consistent throughout in relation to activity and diet, at least at the minimum. This is the influence of diet on urine. So plasma is pretty well controlled by changes, but even the urinary changes here after feeding 10 volunteers diet with saturated fatty acid, monounsaturated fatty acid, and polyunsaturated fatty acid. 4 hours after feeding the diet, you can see how the PCA plot is able to differentiate what diet you gave. So if the diet needs to be standardized for your experiment, it needs to be done across the board. So what are the effects of different sampling collecting strategies? Kelly will talk about this in more detail in the next talk, but I want to briefly touch on what small changes to your study design does to the metabolome. Here we are hoping to capture the cell metabolome, and the decision to detach the cells by quenching the metabolism with different solvents, in this case liquid nitrogen, solvents at -80 and 0 degree. As you can see, this is the glycolytic pathway, and you can see the differences that you can see in the same sample by different quenching methods. So you have to decide what you're hoping to capture in your experiment, decide on one quenching optimal method, and stick to it throughout the experiment. Also, whether you're going to scrape the cells out, do enzymatic digestion. With centrifugation speeds, stick to one centrifugation speed. As you know, higher centrifugation speeds cause cell lysis, and you might get different answers that kind of cloud your answers at the very end. What about the tubes you collect human samples from or mouse samples from? So in this study here, I want to illustrate that importance of what you select as a tube and what kind of coagulation method you use. Here they took 10 healthy samples and pooled their blood and decided to use EDTA, citrate, sodium fluoride, lithium heparin, and serum tubes to cause anticoagulation. And they used albumin blanks as their control. As you can see, the chromatogram yields different patterns And if you pay attention to the lithium heparin aspect, you can see that the polymers of heparin and polyethylene glycol seem to be increasingly causing clouding of the question that you want to answer. So review of literature and our own experience says that EDTA plasma tubes are better if you have a choice to use things. If you're going to use a study that has already been completed with one of these tubes, as long as your study is being done entirety with the same tube, we can still control for these changes. How about hemolysis in your samples? As you all know, obviously, hemolysis increases the free hemoglobin in your sample. It has correlations with AST and potassium in your sample. When we did hemolysis experiments, this is the published literature here I'm showing where the controls without hemolysis are marked in gray, and the blue shows a strong hemolysis, and the red shows a moderate hemolysis created by aspirating up and down. You can see that the PCA plot is able to differentiate the same sample because of the level of hemolysis. Up to 69 features change. Mostly lysophosphocholine 16 and 18 show a strongest association with free hemoglobin. So you have to make a decision to eliminate hemolyzed samples or create some sort of data entry to control for those changes. How do you store and optimally handle samples? What does that happen? What does it do to the question you're trying to answer? So here I'm showing an example of urine that was sitting on the bench for 24 hours. This is an NMR spectra. But as you can see, the same urine sample preserved well on ice versus a spoiled urine sample, you can see new peaks appearing of TMOA, TMA, and acetate. Therefore, temperature changes does cause changes in what's growing, especially in urine, though a sterile specimen originally has to pass through different tracks to become contaminated with bacterial growth. One strategy to prevent the bacterial contamination is to add sodium azide to prevent bacterial growth in your urine. Other effects of temperature, this is an example of 10 serum from healthy volunteers. Here we're specifically measuring hypoxanthine and sphingosine 1-phosphate over time. This is left on room temperature. As you can see, as the time goes on, certain metabolites increase because of cell metabolism or cell decay. So your decision not to maintain these samples in the right temperature could change what you're trying to measure. So always work on ice. Transport samples on dry ice and store them at -80, and if possible limit processing time to the very minimum, and do that across the board so that you are not changing the answers that you're getting. This is the effect of freeze-thaw cycles. We all store samples in large aliquots and we freeze-thaw it many times to use for different assays. This is an example of, again, healthy plasma measuring carnitine when it's fresh and freeze-thawed once, twice, 4 times. As you see, the level of carnitine changes quite a bit with every freeze-thaw. So the effective strategy to prevent this is to make small aliquots for different purposes, or uniformly have the same amount of freeze-thaw cycles throughout, and use as much as possible fresh or one-time freeze-thaw cycle samples. So I hope I've convinced you that timing the sample collection on the same day at the same time would be an effective strategy to prevent changes in your metabolome. Always collect at the same time in relation to a meal or a physical activity. Standardized diet, if not, at least get good diet histories. Standardized collection strategies, and keep samples frozen and limit processing time. So I will conclude by saying that a good metabolomic study will answer all the questions that you have if you standardize and consider consistency throughout sample collection, storage, and data pre-processing. And if at all, if I convinced you with anything over this half-an-hour talk, hopefully it is consistency and consultations before you actually embark on your metabolomics experiment. At this point, I will stop. These are the references I used for the data I was trying to convince you. I will stop to answer questions. Yes?
In regards to sample size, I know you discussed using a pilot to to determine your effect size. But if you're doing an untargeted study where you're not, you're not quite sure what your exact endpoints are that are going to be important, do you have any other strategies for advising people on how many samples?
The strategy always has been a discovery and a validation cohort. So that's the only strategy. Because it's untargeted and we are blindly measuring a lot of variables, we have very little a priori knowledge on what's the difference going to be. Once you know the difference, then they have a lot of strategy to handle it. So targeted methodology as well. Yeah. Yes.
Can you tell me a little bit more about a serum versus plasma, and maybe platelet-rich plasma or pro-plasma? How much of the difference those platelet-derived factors affecting metabolomics results?
Obviously, with serum, you are discarding a coagulate that is including all the platelet activating factors. So if that is the question you're trying to answer, I would use plasma. Yeah. Depends on the question you have, whether serum will answer it or plasma. But whatever the decision you make, the point of the design of the study is to stick to the same thing. If you change midway, your answers are going to be completely different. Yeah.
So you see often market difference?
Not in everything, but certain things definitely, especially the things associated with the clotting aspect will have changes. Yeah. Yes.
How specific is the measure of metabolites in different tissues? For example, if I'm studying Alzheimer's disease, I want brain tissues, but nobody's going to donate their brain. You go for CSF and plasma. And if you are studying pancreatic cancer, you're going to do something else. So given you're going to be working with surrogate, how do you guys deal with how well that measure approximate the target organ that you are looking at?
Without measuring the target organ, it's difficult to achieve, but you can always match the phenotype that you're looking for, the surrogate sample that you're looking.
What do you mean?
For example, if you're studying Alzheimer's disease, you will try to match the clinical phenotype that you're looking for to your surrogate biological fluid. So we are limited with the samples that we can work with. Yeah. Yes.
How do you maybe actually you could just say whether or not in the future you're going to tell us about the way to get rid of all of the large molecules that are absolutely not really interesting. So like proteins and nucleic acids, you don't really want those in there because as you're analyzing for amino acids, that's going to change, affect or kind of contaminate, like your whole proteins and stuff are going to contaminate the metabolites. So you kind of want to get those out. But if you precipitate with TCA, now you have a bunch of TCA in your sample.
So we address that by sample extraction strategies, and depending on the metabolite you want to measure, there are different sample extraction strategies, and if there is also ways to test different sample extraction strategies to match the question you're looking for. If you have a panel that you're looking for that's already established, we have tested it out already in the core, and we know what sample extraction works. But if it's an untargeted kind of thing and you focused on one aspect of the metabolome that you want to measure, we can devise specific sample extraction strategies that focus on that as well. So it is based on sample extraction, which people address later on in this workshop. Yeah. Yes.
A question about the data on normalization. So we do a cell metabolomics study. We have two ways to normalize data. You can use serum counts, you can use protein concentration. So which is the best way to normalize cell counts?
So traditionally for cell culture experiments, we have used cell counts as a measure. But protein concentration is also a possibility. So both are valid ways to normalize. Depends on the question as well.
Can you give examples? What kind of questions?
So for example, if you are looking at changes in amino acids you would basically have protein concentration as a measure of normalization. But if you're looking at growth-related aspects of cell studies, then cell culture, cell count at the beginning where you seeded it makes an importance. So it depends.
So let me just, I can just say the way we usually do it is, usually you'll give us a cell count. We'll do a protein assay at the end of it after extraction. So we can, if there's some loss from the cell count into the extraction, then we can at least adjust for that. But usually it's a pretty reproducible extraction from tissue culture cells. Now, if you get into, spheroids, people are growing, suspension cultures and things like that, it's a little bit more difficult, and then usually rely on protein concentrations a lot more. So, so there's not a great answer. What I always say is that if you're going to measure one thing in your cell, you know how you normalize things. We'll look to you on how it might be better given the characteristics of your cells as well. So remember, there's nothing really special about metabolomics except for the number of metabolites and things that you can get. But a lot of times you'll break open your cell and you'll measure glucose or something like that in it, right? And so, it's essentially the same thing. It's going back to what I was talking about is the extraction methodology, the precipitation and things like that we do. We hope it's reproducible from culture to culture. But sometimes the cells have different characteristics after you do a transformation or things like that, and then we have to work with you to really understand exactly how we can do it. We've been working with an investigator looking at different extraction methodologies and normalization methodologies for different parts of Drosophila's bodies, and it's not the easiest thing to do, especially when there's really small sample volumes.
Yes.
How important is it to race and sex match your samples, and is there a known variability for African Americans versus Europeans for certain metabolite panels?
Sorry.
Okay. All right. How important it is to sex and race match your samples, and is there a known difference between different races and certain metabolite panels?
It's very well known that gender plays a lot of changes for many metabolites in the serum and plasma. So yes, it is very important to well match for gender. Race changes have not specifically been studied, but I'm sure there are changes based on that as well.
There's at least initial with some lipids. There's actually some African Americans tend to have more desaturase activity in their liver. It may be related to the propensity to fatty liver disease and things like that, among other factors. And so there are subtle differences. I don't think we've systematically looked at it. There's clear sex differences that we can find in the lipidome, in the steroid panel, obviously. We recently did a ring trial where we actually had unknown samples sent to us, and we were asked to sex the samples, as it were. And obviously steroids help a lot in that regard. But we could see separation with some of the other platforms as well.
Also age. Age seems to be a big factor as well. So you need to really match your cohort and control to not be answering questions that you don't want answered.
So do you have an idea of the best time point to analyze metabolites, for example, from tryptophan when working with cell culture?
If you tell me, that's my answer. If you tell me a little bit more, then we could probably discuss about what you need. When you do a time course experiment and you're doing a labeling experiment, or if you're doing an intervention experiment, it's really going to be, it's going to depend upon what you're looking for, and how big of a change that you expect. So, but that's why we want to discuss experiments with you before you actually perform them. Otherwise, if you give us that one that's 2 minutes apart, you're not going to see anything. If it takes 2 hours to label it up and things like that. Sometimes you just have to do preliminary experiments just to see exactly what you're going to need to do. So it depends. So I want to just get back to one thing. Maureen, tell me if I'm wrong. So we do have an estimate of the standard deviation of most of our assays online. And so depending upon the matrix that you use and things like that, we can give you an idea of what our variability for specific assays are, mostly in the targeted. The untargeted assays, unfortunately, really depend upon the metabolite that you're looking at.